Hello, everyone. Great to see you here at this Cold Spring Harbor Laboratory event. My name is Steve Mirsky. I was an editor and the podcast host at Scientific American for more than 25 years on the editor side and about 15 on the podcast side. And right now I'm semi-retired, and it's my pleasure to be moderating today's event on the history of adenovirus research at the Cold Spring Harbor Laboratory. And we're gonna be hearing from Terry Grodziger and Bruce Stillman. And before we do any of that kind of interaction, I think it's important, if you're wondering why is the field of adenovirus research important, well, just one example is that some of the vaccines that are currently being distributed for COVID-19, or I should say against COVID-19, are based on adenovirus research and use adenovirus as the vector for the vaccine. So it couldn't be more current, really. So now let me get back to who the participants are in the panel. Terry Grodziger has been at Cold Spring Harbor since 1972, is that right?
Yes, I came at the very end of 1972. Can't believe it myself, but it's absolutely true.
And you have been the editor of Genes and Development since 1989, was it?
Yes.
Yes. So tell us about this, as you called it, a kind of circuitous route that you took to get to the laboratory.
Well, I think the circuitous route I took was actually followed by many people. Or I should say, there were really, everyone who came to the lab, when I did and for years after, we were all young, we were all scientists. Some were biochemists, some were geneticists and cell biologists, but we all had the same goal, which was we really wanted to understand how genes were regulated, how replication of the DNA occurred, how proteins were made. All these basic processes were actually made and carried out in human cells by human genes, or at least mammalian ones. And this was really a black box and very difficult to approach. This was before cloning. It was a transition. And there were several of us, in fact, who had worked on these kinds of questions with bacteria. And we were kind of really interested in guessing. This was true of me and several other people who came guessing that it was worth a try to work on these systems, and that viruses were the best way to do it because they were much smaller and we hoped to approach it. And in fact, at Cold Spring Harbor, there was research going on two viruses. One were a few types of adenoviruses, which the ones we did were human viruses. The ones we worked on were called Ad2 and Ad5. They just have boring numbers. But they caused colds. They were very common, at least in the area we were. And most people, 2/3, 3/4, had antibodies. In fact, when I first came, after a year, we were tested. I think the idea was to see how neat or sloppy our work was. But I had found this article before they finished it, that in fact, 2/3 or 3/4 of the people anyway had antibodies, which is how it turned out. And you'll hear a lot. You saw a picture of our colleague Bob Tietjen, and he had also a biochemist who worked on bacterial proteins, and he came down to exactly see if he could do these same things with human viruses. I should say there were really two viruses. One was called SV40, which just stands for simian virus 40, which was also studied. It was a monkey virus, and it did cause tumors, which is what I think got people at NIH interested in this virus. And the questions were really the same. So people came down. I will say this, there were very few, easy reagents already made. We had to really develop procedures for this. So that's what, there weren't kits, there weren't core facilities where you sent off your mutations and they'd be sequenced overnight. There was nothing. And they were developed, it was hard, and so people's work was as far more specialized as far as techniques were concerned. which really led to lots of collaborations, which was people talked to each other all the time, they collaborated on experiments. And I should say, just in general, the lab was very small, everybody was young, everybody was enthusiastic about their work and talked all the time. I told you before, it was like a scientific commune. That's really what it was like. Everyone lived on the lab grounds, they ate in the main dining hall, and as most communes were then, it was poor. Okay, it was broke. If you really want, which I just mean broke, that it had no endowment. Although several years later, Jim did raise the money for the first endowment, which is called the Robertson Fund. I don't remember if Bruce already was there, but what we did have, which was amazing, was we had a very big, by those standards, grant from the National Cancer Institute. It was called a Program Project Grant, and it supported a lot of different people. It supported salaries, it supported reagents, it supported some equipment. And when I came, what was great is you just arrived. It supported half the, and you got onto this grant. And there it was, and you were set. And I should say, this grant exists to this day. Bruce, it is, I think, the longest existing program project grant in NCI's history. And there were other people who also wrote grants to NCI, and honestly, it was not the tortured procedure it is today. I mean, it was a lot of work to write a grant, but if you were doing good and innovative work and the reviewers thought so, you would get that grant.
Let me introduce Bruce before we go any further into the details of what was going on back then. Bruce Stillman, President and CEO of the Cold Spring Harbor Laboratory. As you'll no doubt notice when he starts to talk, he is from Australia, but came to the lab as a postdoc in 1979 and somehow never went back. So how did all that work?
Well, yeah, so I came here for 2 years, and 41 years later I'm still here. I actually started off wanting to do medicine, and then I got interested in research, and I decided that I wanted to work on cancer. And the viruses that Terry mentioned, adenovirus and SV40, were called DNA tumor viruses. They caused cancer in rodents. Even though adenovirus is a human virus, causes the common cold. Fortunately, it doesn't cause cancer in humans, but it does in rodents. And so I decided as a graduate student that I wanted to study viruses that cause cancer. And I did that at the Australian National University. Did a PhD in about 2.5, 3 years. And then came to Cold Spring Harbor because, as Terry mentioned, it was the place to be for young people. And I brought my research that I started as a graduate student here and focused on how our genome is copied, how DNA is replicated. And I first started that using adenovirus. And then later SV40 virus, and then later yeast, and then later human cells, which we now work on.
Let me also just quick piece of bookkeeping. Anybody who's watching, if you have any questions, you can use the Q&A part of the Zoom to put your question in, and we'll start taking questions at some point during the conversation. So we were talking before we went on the air. And you spoke a little bit about this, Terry, but we're talking about how if you're 25 years old today, you grew up in a world where if you want a genome of a rabbit, you ask for it, and the next day you have the rabbit genome. 20 years ago, 22 years ago, we were at the end of a very long effort to put together the human genome, and now I can have my genome sequenced for $1,000 and I'll get the results pretty quick. But that's a whole different world from where you folks were when you arrived at Cold Spring Harbor Lab and had to you're baking the cake from scratch way back then. So again, you decided you both wanted to work on adenoviruses because you were interested in these very basic cellular, well, not even cellular, but DNA mechanisms, genomic mechanisms, I really should say. So, what was it like back then, and what was it in particular that you were trying to figure out, and how did the adenovirus become your, I can't even say organism, but your system for doing that work?
Well, I should say I started, as I say, people specialized. I was the geneticist in the building I was in, and there were lots of collaborations trying to, well, of course, it was Barbara McClintock. I should add the fabulous geneticist Barbara McClintock, who was always great to talk to.
Jeans lady up at Cornell.
Well, she was at Cold Spring Harbor. She had moved to the Carnegie Institution of Washington, which had a department of genetics, I think it was, at the lab. And they had then, Al Hershey was at too, they had pulled out, contributing to the brokenness of financial travails of the lab and later fundraising. But I had worked on bacterial genetics and isolated several different kinds of mutations. And they were the most basic questions first. I was going to make mutations, analyze them, also with colleagues. We'll keep mentioning people. They were basic questions like, did you get the same kinds of mutations and mutagenesis range in human cells and in viruses that you did in bacteria? Could we map them onto the DNA of the small viral chromosome? And these, again, were collaborations. This project with all the mapping, you saw even I was the geneticist, Joe Sambrook and James. Well, you saw a picture of the back of Joe, of his head. And Phil Sharp was at Cold Spring Harbor before I left, and they would analyze the DNA of this. And this really helped a lot, knowing where things were for this vector project, we'll say. But there was I'll just say another thing with the genetics of going on to vectors is there's also serendipity, and this was not us. But at NIH, they were growing loads of SV40. I'm not sure, there were several stories of why. Were they trying to make vaccines or antibodies? But they also had adenovirus around, and somehow one of the cultures or several got contaminated with adenovirus. And talk about evolution, one of the most important pieces of SV40 got crossed into or inserted into the adenovirus genome. The virus grew fine because it took out a gene that was not essential to grow in tissue culture, but it had this region of T antigen, which I'm sure you'll hear about more from Bruce, that was involved in, it was a real control protein. It was involved in replication, it was involved in gene regulation and transformation, and what it did is it allowed this adenovirus that only grew in human cells to grow in monkey cells. So it shows you what both evolution and serendipity can do, because this became an incredibly useful piece of DNA, both for studies, because you could kind of select for and do clever genetic tricks, but it also sort of inspired the search for the people for making vectors. So let me say, all these interesting proteins are made in very small amounts, which is perfectly okay if you're doing the genetics or DNA, but it's very frustrating for the biochemists who really want to get hold of these proteins, purify them, and study their functions. So there was a real impulse, for me to really see, could we make vectors that would grow in human cells? And we had a head start as a proof of principle because we had this piece of SV40 that was expressed just as miserably as it was anywhere else. But was it a proof of principle that we could, have genetic tricks to try and move it to a place? And all these basic studies had showed that there was one control element in adenovirus that really ran things very efficiently. So could we use some tricks to get that T antigen gene over in some way to be expressed by this high element, and it would way overproduce T antigen, which would be then easy to study. And this part was a collaboration between me, who was, I think, a very good geneticist, but I knew biochemistry, but believe me, I couldn't do it, and Bob Tejan, who had come from Harvard, where he'd done biochemistry on bacteria, who knew all about genetics and didn't want to do it, but was a really, really good biochemist. So this was a joint project. It was a proof of principle. Could we get this to work. And I will just say most of this work was in fact done quickly at Berkeley, because we started at Cold Spring Harbor. Teach then got a job as an assistant professor at Berkeley, and somehow the rules at Berkeley for handling viruses were not as strict as on Long Island. I mean, they were strict on Berkeley and Long Island, but don't ask me what happened in New York State. I've repressed why they might have thought this. So it started. One of the first really cross-continent collaborations. I would go and visit Berkeley, spend weeks there every year. There was a wonderful graduate student named Carl Thummel of Tejas who worked on this project. And it was a kind of collaborations that went on in these fields. You would get reagents from people at other places too. Everybody was very generous with exchanging things. So that's what happened. And of course, I wouldn't be telling you about it. I don't know how much was clever and how much was just luck that this worked so well, which then inspired us as more proof of principle to try all kinds of different genes.
You know, I have to bring up the famous Louis Pasteur quote that chance favors the prepared mind. So you might have had serendipitous events occur, but to be able to realize it and harness that is a testament to the actual talent.
My final word on this subject, which is on the other side, is someone said, you know, if 20% of your good ideas work out, you're really lucky. This is because evolution intervenes between logic and carrying things, how things get carried out.
It's going to do that. Bruce, let me ask you, what were some of the challenges that you faced in changing wild type adenovirus into something that you could use in these situations? I mean, you can't inject adenovirus in somebody if it's just going to multiply all over the place. You want to make sure that it goes in and doesn't do anything that you don't want it to do.
Yeah, so the Cold Spring Harbor Laboratory was really at the forefront of understanding the structure of the genes within adenovirus. Just as a comparison, in our genome we have 3 billion base pairs of DNA. Adenovirus has 40,000. So, and within that 40,000 nucleotides of DNA, there are a bunch of genes that were all mapped here. And one could delete those genes, and some were essential and some were not essential. But there was an important breakthrough. The cancer-causing genes, which is what a lot of people were really interested in, you could clone those after recombinant DNA came into existence, which was in the mid-1970s. You could clone these cancer-causing genes and put them into human cells. And those human cells would proliferate forever. And there was a famous experiment done in the Netherlands in 1977 where these cancer-causing genes of adenovirus were put into a human embryonic kidney cell. And that cell line is called 293 cells. It grew in plastic dishes. And that means that you could infect those cells with adenovirus that had deleted the cancer-causing genes, and it would replicate because of genetic complementation. So a scientist who worked with Terry and myself, Yasha Glucksman, decided that he would put foreign genes, replace the cancer-causing genes, and would put foreign genes into adenovirus. And this became the first what we call helper-independent adenovirus vector to produce a foreign protein. And it had to grow in these 293 cells. Now, my laboratory was interested in expressing proteins and doing biochemistry. And we worked with Yasha to express the full-length SV40T antigen in adenovirus in a helper-independent manner. And we had to grow these in 293 cells. But they grew in these plastic dishes, which was very cumbersome to do. So I decided that I would force the 293 cells to grow in suspension culture so we could grow them in hundreds of liters at a time. And it took about a year to do that. But those cells which I used for many, many years to do biochemistry. It turns out they're the very same cells that these adenoviruses are used for making these vaccines today. And because you can grow them in very, very large amounts, you can infect them with these recombinant adenoviruses that express the spike protein of the coronavirus that's causing the current COVID pandemic, and thereby make lots and lots and lots of virus, which becomes the basis of the vaccine.
You
So
skipped over.
making those cells, in the early 80s, I had no idea that they would be important, down the road in making vaccines for a pandemic.
But you skipped over a really interesting, to me anyway, an interesting aspect is when you're growing out the adenovirus, when you're learning how to make it grow in the flask, in the solution, rather than in the petri dish. That's a laborious process, but you're also taking advantage of evolutionary principles. And could you just, for a minute, just talk about that a little bit?
Well, once all of the genes were mapped, which was done by people at Cold Spring Harbor and elsewhere, once all the genes were mapped, you could. And the other thing about Cold Spring Harbor is because of this collaborative nature, there was a scientist here, Richard Roberts, who went on to win the Nobel Prize for work on adenovirus with Phil Sharp. They had isolated all these enzymes that could cut up DNA and make recombinant DNA, and those were available here. So it was very easy then, after recombinant DNA came along, to clone various genes into DNA and then design them so that you could put them into adenovirus. So this was a trick. As I said, Terry first started doing this kind of evolution, showed you could do this by what she explained by fortuitous recombination. But later on, we could do this deliberately and express any protein in adenovirus that we wanted. And so that was really, I think Cold Spring Harbor really made that available because we had all of these enzymes that you could just go to the freezer of Rich's lab and get all these enzymes that you could make recombinant adenoviruses. And then, as I said, making the cells grow in suspension enabled the production of very, very large amounts of cells. We would grow routinely hundreds of liters a week of human 293 cells, and either infect them with adenovirus or not, and make extracts and do biochemistry. So that was what I did for about 12 years, and got a lot of really interesting observations out of that.
Very good. Terry, you mentioned working with Tej at Berkeley, and there's a kind of an interesting story about your attempt to patent what you guys came up with.
Well, yes. Let me say, once we had shown that, in fact, just to say that these viruses that expressed T antigen in this way, they overproduce the amount of protein then that you would have gotten by 20 to 40 fold. So that was the level. So we thought, and we wanted to make it, obviously, as I said, in human cells, so also the proteins would be properly made and modified. Just to say that proteins in cells they're a string of amino acids, as you know, but they can have many chemical groups attached to the proteins that can affect their activity for good or ill, its stability, make it more or less stable. So it was really important. One reason we wanted to make all of these inserts and genes in human cells was that they would be biologically active. So yes, we thought, well, no one's done this. This is really novel that we have this method for inserting genes. And let me say, Cold Spring Harbor, I don't think it had any office to patent it because, as Bruce said, Yasha had developed cells called COS cells, which had SV40 T antigen in them and could complement all kinds of mutants. And he shared these with everybody, I will say. And he thought about patenting it then. I will say Yasha was from Russia. Then he moved to Israel and he came here. And he was probably the most enthusiastic capitalist, given his childhood in Russia, of anyone at the lab. And he thought about it, but there was a rule, which may still exist, that if you talked about it publicly in any way, you had only 1 year to submit a patent. And so that didn't work out and loused it up. Okay, so Cold Spring Harbor wasn't so good. This is before biotech companies, before anything. I will say, Bruce didn't say with Rich, the lab or Jim were not interested in companies in this. So off at Berkeley, it was big, and we thought, okay, we'll submit a patent application for this novel method of producing things, which we did. Tej and me, and we put Carl on the patent. Anyway, off it goes, and it comes back and it says, no, it's not novel enough. And we go, oh, really? Okay. That was the end of it, because we had no place else to fight. Well, at Cold Spring Harbor, which wasn't interested in business, shall we say. They did become so very effectively later. But let me say, the initial scientific experiments were much more successful than the patenting attempts.
How many billions of dollars were left on the table there, do you think?
Oh my God, Bruce's would be better at taking a guess.
Well, actually, to be honest, I think we wouldn't have benefited at all from those patents, because the patents would have issued in the early 1980s, and it wasn't until much later that people decided to use adenoviruses as vectors for either gene therapy, which didn't work, or for vaccines. And so the patents would have expired by now. But I think what's critical about this is that the very basic research that was done here and elsewhere on adenovirus, fundamental research to understand how genes are copied, how genes are transcribed, and how proteins are made in cells. All of that was really driven by studying of these viruses. And it was fundamental research that was necessary to as a precursor to understand how our cells work. And so this fundamental research ultimately led to people having the idea that maybe you could use adenoviruses as vaccines. And so the two vaccines that exist now for COVID SARS virus are a J&J virus that is actually a different adenovirus. It's a different type of adenovirus from humans, because, as Terry said, we all have antibodies to common adenoviruses. So they picked an uncommon adenovirus, it's called adenovirus number 26, that people didn't have antibodies against, and they expressed the spike protein from SARS virus in that, and that's the Johnson & Johnson vaccine now. The group at Oxford, because of the antibody problem, actually chose a chimpanzee adenovirus, and that's the AstraZeneca one that is actually giving a lot of controversy at the moment. But both of them are recombinant adenoviruses. They use the same technology that was worked on here in the early 80s, exactly the same. They used the cells that were, as I mentioned before, grown in large amounts here. And you can purify these adenoviruses. They're very robust, and you can purify them very cleanly, so that when you inject them into people as a vaccine, they don't cause any side effects or problems, except the side effects that are caused by the SARS spike protein, which many people who have been vaccinated will know that there's an immune reaction against that protein.
Right. I want to it occurred to me that people might hear chimpanzee adenovirus and think they're getting something from a chimp. No, it's a virus that infects chimpanzees, and we're getting that virus. But the virus itself has no effect on us. It's just used as a carrier so that our cells will produce the spike protein that will alert our immune system to go get it and protect us from COVID.
It's like a Trojan horse, and in fact, when when these cells are infected by the recombinant adenovirus, those cells are eventually killed. And so there's no long-lasting effect of any of these in our body, except that you gain immunity against the SARS virus. And that is so that they're actually very safe. They have been used quite a lot for different types of vaccines. But the technology nowadays is such that you can make these recombinant viruses very, very quickly. So, for instance, all these different variants of coronavirus, the SARS coronavirus that are emerging, like the British one and the South African one and the one from New York and New Jersey, those variants that can be very easily cloned into adenovirus, and you can make a vaccine against those as well. So it's
you've got the basic vaccine, you can make alterations in it almost absurdly quickly.
Yes, very quickly. I mean, you could actually make one of these vectors easily within a month. And then you have to obviously, because it's going into people, you have to do clinical studies and all of that. But the actual production of the virus will take about a month.
Which is really important. I've seen people. I saw somebody on television say that they were concerned about taking any of these vaccines for COVID because the researchers developed them so quickly, and she didn't trust these things because they were developed so quickly. But here's an explanation for why you can develop some of these things so quickly because of 50 years of groundwork. It's really, an overnight success after 50 years.
Yeah, but the other thing about quickly, normally vaccines were developed against mumps and measles and other infectious agents, and they would take a number of years, 4, 5, sometimes longer years to develop. And that is because there was time to do that. When you have a global pandemic like the SARS-CoV-2, COVID-19 virus, it was imperative that we actually try to shut that down quickly. And you can see the consequences of not doing it. I mean, many, many people in the United States died. So there was a need to rapidly develop vaccines. And all of the technologies that were developed here and elsewhere of cloning DNA, making recombinant viruses, even making messenger RNAs, which are the bases of the different vaccines from Pfizer and Moderna. Those technologies all existed and can be very rapidly employed. And I think it's very important. Now, the clinical trials were done, and it is very important that in new virus vaccines, like the adenovirus vaccines, that you have to show that they're safe. And that's why it took a year to make these, to get them approved on an emergency basis, because it takes some time to demonstrate that there's no long-term, many months of side effects from these agents.
I'm curious, from an evolutionary standpoint, adenoviruses are poodles, but were they ever wolves? I mean, in our interactions with them, were they ever much more dangerous than they are today?
Well, part of the reason they were studied is that adenoviruses typically cause the common cold. They're one of many, many viruses that cause colds. But in the 1950s, there were, especially when a lot of soldiers were billeted together in army huts and things like that, these viruses actually spread very rapidly through populations. And there was severe outbreaks of adenovirus that it didn't cause death, but it caused a lot of problems, respiratory problems for soldiers. And that's one of the reasons why they were first studied. Then later on, people found that when you inject these viruses into rodents, they would cause cancer at the site of injection. They don't do that in primates or in humans. but in rodents they do, and this then led to the interest of, what are the cancer-causing genes in these viruses? And that's why they were studied.
I should say also one of the features with viruses of these viruses, because they're growing, when you can try, is that it's really possible, which we can show, to put in, this was part of a proof of principle, to put in proteins where you could really show easily. It was important, are they functionally active? After T antigen, you could put in, these were all done, as Bruce said, because you could clone these things on bacteria. And so you could take an active promoter, an SV40, and you give it to the virus. They like to recombine. And at some level, they'll kind of grab the same sequences that are in this piece of DNA you give them and insert them into the virus, just in the way it happened in this unplanned accident at NIH. So then, one could take, and we could select for them. That was the gift and the trick. But then you could try just putting things between them. The promoter, you wanted to make an enzyme, and put the SV40 in there. And by always using this monkey cell, you could select ones out. So we could produce, we tried an enzyme that came actually from herpes virus, which was cloned. And it was another example, we got them, people gave them to us. And we tried the equivalent, another related to T antigen mouse virus that made a complex set of T antigens. And it managed to do that and overproduce them. And the last thing we did, I honestly forgot about this because I think we weren't allowed to do too much, but we actually got one of the first cDNAs for a human gene. It was for a hormone. This was one of the visiting postdocs in my lab who worked on retroviruses and went on, his name was Roger Cohn, to have a really good career working on viruses. They were very rare. Bruce may remember this clone. It was a subunit of a human hormone called chorionic gonadotropin. And we just put this into this system. It got made, and we could show that, like I would say with herpes virus, it was active. This we could show that it was glycosylated. It had sugar units added, which is what it did, and that it was secreted from the cell. I think that was the end probably of what we were allowed to do with this. So it became another proof of principle and is more relevant in also to the gene therapy aspects of using adenovirus. But they were just versatile, wonderful things if you decided to give it a shot.
So,
so to speak.
so you just mentioned gene therapy, and that is another potential avenue for the use of adenoviruses in gene therapy.
Yes, I have to say, I think for the first it was about 5 or 6 or 7 years after I saw one paper from some big pharmaceutical company that I can't remember that was trying to use adenoviruses as a vector for something. That was it for a long time, as Bruce said and described.
I think that the adenoviruses are not going to be used for gene therapy. There are other viruses that are used now that are much more effective for gene therapy, although gene therapy has still got a long way to go. Nowadays, with the CRISPR editing, gene editing, I think that's going to bypass a lot of the use of viruses for expression, complementation of genetic mutations, for instance, in gene therapy. So this CRISPR technology, I think, has now taken us to a whole different level that we won't be using many viruses. There are viruses now that are being used for instance, treating human disease. There's a disease that called spinal muscular atrophy, for which a gene in a recombinant virus is put into infants, and that has some effect, and that's been approved. But I think the CRISPR technology is going to bypass all of this now.
I'm looking at some of the questions from folks in our audience, and I know the answer to this, but I'm going to ask it anyway because some people might be interested in the answer, and that is, if you could choose which vaccine you were going to get, would you choose one over another?
Well, I would take what vaccine I could get hold of, because the reality is The SARS virus is a pretty dangerous virus. It can cause death, as we know. And what's interesting is you can't predict who's going to get very severe symptoms and who is not very easily. And so if you have the opportunity of getting vaccinated, I think the 3 that have been approved in the United States, the Moderna and Pfizer messenger RNA vaccines, or the J&J adenovirus vaccine, are all equally effective in preventing severe disease and preventing hospitalization. So I don't think it really matters. If the J&J one, I think, is going to be, which is the adenovirus one we've been talking about, is a single shot vaccine. It could be, you could get a booster from a second shot, but at the moment it's approved for a single shot, and that is effective in preventing hospitalizations and severe disease. And I think it's the one that's scalable into very, very large amounts. This is the vaccine, I think, that could go into the into third world and is going to other countries and being distributed. The messenger RNA vaccines are very effective, but they're a lot more difficult to make and transport and use.
I would say I would totally agree with that. I think I certainly would have been and was happy to take whichever one was being given at the place I was getting it. I think they're all very effective. I mean, I think Bruce makes a good point that the J&J ones are not only easier, they can be used in more places, they're much cheaper because they don't need all of this freezing and super freezing. They can just be refrigerated. It's almost more in principle of ease and cost of getting a flu shot. I mean, you can walk into many places. People don't need a lot of advanced training to properly do the injections. You know, they need special syringes. But I think that they're all really very effective, and anyone should feel it fine about getting any one of those.
Yeah, you see numbers, lower numbers for the adenovirus vaccines in terms of efficacy, but in terms of preventing severe disease and death, they're all pretty much the same. Is that correct?
Yes.
Yes.
And even, just because you've been vaccinated doesn't mean you can't be infected. But what if you have been vaccinated with any of these, the virus will have a very limited ability to replicate in someone that's producing antibodies and ultimately T cells against the infected virus cells. And you very much limit the progression of the symptoms. And so I think that's going to be, and even with the variants that are cropping up, those are somewhat protective as well. So I think we're moving. The paradox in the United States is that the United States was probably the worst country in dealing with the pandemic in terms of how we approached limiting the spread of the virus. But we're going to be the first country that will have herd immunity, I suspect, as long as everybody gets vaccinated, or as many people as possible.
Yeah. I would say people shouldn't mind talking about something easy. We talked about flu shots, and nobody thinks anything about that you need a flu shot every year. The flu vaccine probably isn't quite as effective, but more, you keep asking about evolution, that when influenza viruses have a reservoir in some animals, they create variants at a much higher rate, at a high rate, which is, it may go for 4 or 5 years. We see where they're very minor and the flu vaccine will work, but why they're always making new flu vaccines, which, as Bruce said, there's technology to do it in an amazing way here, is because you now have flu viruses with different sequences and mutations that may affect their infectivity differently, which is why there are new flu vaccines being made all the time. And people go for boosters every year, and at least we are happy to do so, and people should be. So it's not an unknown in principle phenomenon.
Absolutely. This is a question from our audience, and it's outside of your areas of expertise, but I'm going to ask it anyway. Do you foresee any ability to predict new variants of COVID-19 SARS-2 virus by observing the current variants and how they have developed? Is there a way that the variant outbursts can help you predict future variants?
Yeah, actually, we know a lot about that. What's remarkable is the pace of research these days is such that, this virus appeared in the United States just about a year and say 12 or 13 months ago. Very rapidly, within months, the virus proteins were cloned, expressed, and the three-dimensional structure of the proteins was determined. And then antibodies were produced against the virus surface proteins, and the structure of the antibody bound to the spike protein was determined. And not just one antibody, but multiple antibodies. And this was done by collaboration, scientific collaborations across the United States and in Europe and even in China. And so now we have the structure of the spike protein, which is the one that you get vaccinated against, and antibodies bound to that protein. And so if you want to predict how you would get viruses that would escape the immunity, you would be able to look for parts of getting mutations in part of the spike protein that would prevent that protein from binding to the antibodies that already exist. And so in fact some of the variants that we know about now, the famous British variant and the South African variant, are actually mutations in the antibody binding sites. And this is why Regeneron, when they made recombinant antibodies that are used for preventing severe disease and are very effective, they made two monoclonal antibodies, not just one, to prevent this escape from by mutation. So I think this is a constant arms race. The virus wants to proliferate, and there's lots of random mutations. We're trying to control it, and it may well be down the road that we'll need an annual coronavirus vaccine, or it may well be this thing will just go like other coronaviruses and become endemic in the population but not cause major pandemic.
I just want to add one point to what Bruce said, which isn't about directly your question, but people shouldn't have the idea, which you might get, that every variant, every new mutation a virus picks up is harmful and makes it more dangerous, because the exact opposite is true. Most mutations have no effect at all, and many can even make the virus itself less healthy, so to speak, and grow less well. Of course, there are variants that make it more infectious, which is a concern, and will take the kind of work that Bruce is talking about, which is getting more effective all the time to make boosters and things. But it's not as if every time you get a variant, it's bad, which is lucky for everybody.
In fact, there's a company that was spun out of Stony Brook University on Long Island that is taking a lesson from the poliovirus vaccines, which are mutated polioviruses that don't cause disease, but they allow vaccination. And this company is making, or has made, what are called attenuated coronaviruses that will actually be used as a vaccine but will not cause disease. And they've got a lot of mutations in those viruses that prevent the virus from replicating very rapidly and also causing disease. And so this is the basis of the polio vaccines that many of us have had, and now could become down the road the basis of a coronavirus vaccine.
Is there any possibility, this is one of the audience questions, of recombination to generate a replication competent adenovirus, or in the case you were just talking about, an actual coronavirus based vaccine?
I think the likelihood is, I mean, everything is possible, but I think the likelihood of that happening is very, very low. And the reason is that these vaccines, that are, the vaccine that's been injected into your arm is making the spike protein, which is incredibly antigenic. This is why people get fevers after getting vaccination. It is unbelievably recognized by the immune system as a very, very foreign protein. And so those cells will eventually get killed. The cells that are producing the spike protein, which have the adenovirus in the genome in it. And so those cells will get killed, and the chance of some time down the road of having another adenovirus coming along and recombining, I think, is very, very, very low. And so I don't think anybody's really concerned about that.
Mm-hmm. Terry?
I have nothing to add. I agree with that, yeah.
There's a question from somebody in a master's program right now. Is the environment to do research in general just a lot more competitive and difficult now because of the idea of just basic research is not as friendly as it was a while back?
I don't know. It was pretty competitive back in those days, and but competitive is not really. I mean, everybody talks about competition. Scientists don't go into science to be competitive with other scientists. They go in to try and solve problems. And there's a competition. I mean, you see this with the development of CRISPR and all of the patent issues that came out of that. But scientists don't think about competition when you're actually doing experiments. You're there to try and solve a biological or medical problem. And that it was the same when I started off at Cold Spring Harbor, and I think it's the same today. There is a tendency these days to want to do more kind of what I call applied translational research, to solve a problem, and rather than do fundamental research. And I think that's rather dangerous if we go too far down that road, because there are just huge numbers of things that could be discovered that we don't know about, and CRISPR is one of those. I mean, there was a bunch of people who were working on this that nobody was interested in funding the work, and in fact, much of it was done in industry because of that. And yet, that caused a revolution in understanding gene editing. And I think it's already had and is going to have an enormous impact on medicine. So I think there's going to have to be a balance. All of us at Cold Spring Harbor are very interested in applying the knowledge that we have to treating human disease. That's one of the reasons why we're working in this area. And in fact, some of our scientists who came and studied adenovirus, one of them has developed a major drug for spinal muscular atrophy that is just a revolutionary drug, keeping children alive who would otherwise die. And that all came out of very basic science. So, I think there has to be a balance here. And so we do need to keep funding fundamental research, and talking about competition. Now there's going to be competition between countries. I mean, when we all started at Cold Spring Harbor, the United States, and the reason why I came to the United States was the United States was the place to do cutting-edge science. It still is, but many other countries, and particularly China, are putting lots and lots of resources into basic science. And this is not just true in medicine, it's artificial intelligence, all other areas as well. So we need to have continued support of fundamental research, which is going to be an economic driver. It's absolutely the case.
Right. Is it more competitive now to get funding than
Oh yeah. Yeah, when, as Terry mentioned before, back in the early 80s, you would write a grant. There was probably in this field, because it was so hot and everybody was interested in it, you had a 60% chance of getting funded. These days, you've got about an 8% chance of getting funded. And it is extremely hard and competitive to get funded these days. And I think to the detriment of science, because it turns a lot of young people off. And that is a problem.
I would say two things. First, about competitiveness, which people are competitive. They were back then when we were doing hot stuff, and the competition was fewer people, but it was just as strong. But people want to do important things and interact. I will say one thing that also helped Cold Spring Harbor, being small, is on the slides at the beginning was a picture of a map of SV40 with where restriction enzymes cut it. That was actually from way back when, a DNA tumor virus meeting that was held every year at Cold Spring Harbor. And if there were 6 labs in the US, 6 labs overseas, everybody came. And to this day, they were encouraged, and everybody talked about unpublished work. That doesn't mean everyone wasn't interested in trying to make great discoveries, but it happened. The other thing I would just say about grants, I certainly don't disagree with what Bruce said, is that there's certainly more people, but also for like the National Cancer Institute, they are giving, and it didn't, a lot of the grants now that are applied are for tons of money, way out of the ballpark of what would be given. And I think that there's one institute that doesn't have a disease or a body part attached to it, which is the National Institute of General Medical Sciences. And they have found a way to adjust their grants, and I think, Bruce, you can correct me, that they're funding at 25%. Now, it's not 50 or 60%, but in fact, it's sort of shocking. How could cancer be funding at lower rates than all these aging or all these other things? And it's how you also decide to allocate the money. And I agree with Bruce, too. It's out of basic research that can come these applied things. And that's who's doing this, that will then turn into, when done well, various kinds of therapies.
Very quickly, because we're pretty much out of time, adenoviruses cause colds. Are we ever going to see a cure for the common cold?
No, because they're not. Do you know? There are hundreds of rhinoviruses. I think there are 139 rhinoviruses that also cause colds. Colds are a very popular area for viruses to attack your respiratory system and give you a disease. Now, maybe with CRISPR, Bruce, there will be a everything kind of virus cold thing. But wouldn't you rather have one for really bad diseases then?
And actually, there are many coronaviruses that people have been infected with and which also cause mild respiratory disease that you would never have known that you've been infected. So actually, I think it's probably good that all of these viruses cause these colds and mild symptoms, because it trains your immune system, and I think keeps it robust. And that, I think, is something which we all need. If we were in a completely sterile environment, we would evolve not to have an immune system, and then we would be in big trouble.
Excellent. Hey, this was great. Thank you both so much. It was a fascinating hour to talk about this history and how we're living with the outcome of it today. It's just great stuff. Thank you so much. And thanks to everybody who tuned in. We'll see you next time.
Great. Thanks, Steve.